is, the actual contraction process involves the sliding of the myofilaments. They slide across each other during contraction. So we will see how that happens. There are two types of myofilaments. These are just called the thick and the thin, which is nice because they're easy names. The thick just means it has a thicker diameter. Okay, and this is made up of molecules that are called myosin molecules. Okay, so it's just a bunch of myosin molecules, and this is basically what one myosin molecule looks like. It has these kind of squiggly looking, this what's called the tail, and it has these two kind of oval-shaped heads. These are called the heads, also referred to as cross bridges. I'm running out of black. Yeah, that's better. Okay, so this is one myosin molecule. And again, the thick filament would be millions of these myosin molecules. These are the heads, also called cross bridges. And this is the tail of this myosin molecule. Okay? The thin filament actually is thinner in diameter, but it's a little bit more involved. There are three molecules that make up the thin filament, and I'd like you to know all of those because they all have a role in contraction, okay? When calcium is released, calcium is going to go out and it's going to bind to the thin filament, and that will actually trigger the sliding mechanism, okay? During that sliding process, what happens is these myosin heads will bind to actin molecules on the thin filament. So actin molecules make up kind of the bulk of the thin filament. Most of the thin filament is actin. Okay, so we will say actin binds to myosin during contraction. We'll take a look at what this looks like in just a minute. Okay, and if we were to take actin and myosin molecules and put them together like in a test tube, they have such a strong chemical reaction to each other, they will automatically form a chemical bond, and they'll stay that way. So we don't want that to happen when our cells are relaxed, when we want the muscle to be relaxed, because if they were always forming a bond with each other, all of our muscles would always be contracted and we wouldn't be able to relax them. So to prevent that from happening, something has to cover the binding sites on the actin molecules, and that is going to be this molecule here. It's called tropomyosin, and it wraps around the thin filament, covering actin's binding sites so that myosin cannot bind. Okay, so we'll just say tropomyosin covers actin's binding sites. Okay, when the cell is relaxed. Now when we want the muscle cell to contract, we of course have to expose those binding sites so that myosin can bind to actin. So that is where the third part of the thin filament comes in. Troponin is a molecule that's kind of scattered along the thin filament, and this is what calcium binds to. Okay, so when calcium is released, it goes out into the sarcoplasm, it binds to troponin. This will actually have kind of a different shape. The troponin calcium complex is what it's called. It's like a bigger molecule that will literally shove the tropomyosin off of actin's binding sites, exposing the binding sites for myosin. So troponin binds to calcium. Okay, I'm going to use the chemical abbreviation for calcium during contraction. So let's take a look at the filaments and I'll go through that again when we're actually looking at a diagram. Mhm.
Oh, just really quick, it binds to calcium during contraction, but what did you say that shoves off the tropomyosin?
I'll go through that again when we look at the diagram. It will make more sense, okay? You'll see that troponin and tropomyosin are located like right next to each other. So when calcium binds to troponin, that's going to kind of shove the tropomyosin over. And then the binding sites on actin will be exposed. Okay, so let's take a look at a diagram that shows the filaments. You guys have this? Okay. Okay, so this is the thick filament. Again, this is a myosin molecule. And you can see the thick filament is just a bunch of myosin molecules. Notice that the heads kind of stick out all around it, okay? These are going to kind of stick out facing the thin filaments so that the heads can bind to the thin filament, and that will cause that sliding process, okay? So let's take a look here at the thin filament, and then we'll kind of see how these are positioned in relation to each other. Okay, so this is just showing the thin filament. We said the thin filament has those 3 molecules. Notice most of this is these little kind of spherical red molecules. This is actin, okay? We said actin forms most of the thin filament, so it's all these little red molecules. And if you look closely, you can see that little dark spot here, kind of peeking out, little dark spot on them. This is what is called either the active site or the binding site. I like to call this the binding site because this is what myosin binds to. Okay, so we'll say it's the binding site for myosin. Now notice that these are kind of covered up. I kind of tried to make them look a little bit more visible so you could see what I was pointing to. But notice they're kind of covered, partially covered here by this long kind of thread-like structure that's wrapping around the thin filament. Looks like licorice. This is tropomyosin. Okay, so tropomyosin's job is to cover these binding sites so that the cell can be relaxed. It will prevent myosin from binding. Okay, as soon as these binding sites are exposed, myosin will bind, and the filaments will slide across each other, and the cell contracts. So these have to be covered when the cell is relaxed. Okay, so that is what tropomyosin does. And it is labeled for you here. Okay, so it covers the binding sites. Again, so this is when the cell is relaxed. Okay, and you can see how troponin, which is this blue complex, is kind of scattered along the thin filament. Notice its close relationship here with tropomyosin. So when calcium comes and binds to troponin, it will change the shape of the molecule here. It forms a larger molecule that literally shoves tropomyosin over. It literally moves it over, so it's no longer on these binding sites. The binding sites for myosin are exposed. Myosin automatically binds. Okay? And we'll talk about what happens when myosin binds. The little heads will bind to the binding sites. Okay? So the troponin complex, as it is called, binds to calcium during contraction. And we'll just add causing tropomyosin to move, and this exposes the binding sites on the actin molecules. Okay? You guys have any questions on any of that? So again, you guys, all we're doing is, as we go through these parts of the cell, we're just talking about basic functions, okay? So when you review this, you say to yourself, okay, what does actin do? Well, it has binding sites for myosin. What does the tropomyosin do? It covers those binding sites. What does troponin do? It binds to calcium. That moves the tropomyosin. And so we're not really going into as much detail as I know it probably feels like we are. So when you review this, just think basic functions, that's all we're doing, okay? Okay, so let's look at how the thin and thick filament are arranged in relation to each other so we can understand how this sliding thing happens. Okay, and this is just a table if you want like a quick review of the parts of the muscle cell. It has all of these parts of the cell listed, and it will tell you what they look like and what their functions are. Okay, so this is, I think, a little too much of a close-up, but let's just use this for a minute here. You can see how the thick filament, again, has, you know, all the little myosin molecules, their little heads are sticking out kind of close to the thin filaments so that they can bind. So as soon as calcium binds to troponin, and that causes the tropomyosin to shift, exposing the binding sites here, the myosin heads will automatically bind. So they're going to make contact here with the thin filament, okay? This little all around the thick filaments, they actually bind. And when that happens, myosin heads have kind of a swiveling motion. And so they're going to literally move the thin filaments, and that's what causes the sliding. And we'll talk more about that when we get into a little bit more of a picture of it here. The reason these heads are also called cross bridges is because, notice they're forming like a little bridge between the thin and thick filament. So that is why these are also called cross bridges. Okay, so looking at this in a little bit more of a big picture, what we're going to take a look at now is something called a sarcomere. So we'll talk about all of this, okay, and we'll come back to this. Okay, so this is a sarcomere. The sarcomere is one unit of myofilaments, okay? That is what it is. One unit of myofilaments. Okay, it goes from what is called one Z disc, okay, to the next Z disc. And what we'll take a look at here is, in this sarcomere, how the filaments slide across each other. So because the filaments will slide, that will cause the sarcomere to shorten, right? It will shorten, the two Z discs will come closer together as that happens, okay? So this is called the functional unit of skeletal muscle because this is where contraction is actually happening, okay? So sarcomeres are the functional unit of skeletal muscle. Okay? This is where the actual contraction process will happen. So let's say calcium has been released. We've had an action potential that will cause calcium to be released. Calcium will come in and bind to troponin. The tropomyosin will move, exposing the binding sites on actin, and now the cross bridges or heads of the myosin molecules will bind. So when they bind to actin, they will pivot and release those actin molecules, then they go back and they bind again, and they pivot and release, and they go back and bind again, pivot, release. So what they're really doing is they're pulling the thin filaments toward the center of the sarcomere. Okay, the thin filaments will move toward the center of the sarcomere, so the filaments are sliding across each other. The two Z discs will come closer together, the entire sarcomere shortens. If you look at this picture, let's go back to this diagram just for a second here. This is a myofibril, okay? The whole thing here is a muscle fiber, one muscle fiber or cell. Okay, so it's showing one myofibril sticking out, and notice that you can see, again, that pattern that causes, the striations in skeletal muscle. You see there's this kind of regular pattern that repeats itself. This is because sarcomeres are lined up end to end, all the way down the length of the myofibril. So notice that if you look closely, you can see all of these Z discs, okay? You guys see the Z discs? I don't know if you can see that detail. Okay, so these are all sarcomeres. So this is one sarcomere here, there's another one here. So because these are connected end to end, as the contraction process happens and the myofilaments slide, each sarcomere is contracting, right? And they're all connected end to end. This causes the entire cell to shorten or contract, okay? This is called the sliding filament theory of muscle contraction, but this has been proven, and this is how skeletal muscle contracts. The filaments slide across each other. So let's go back to our picture of a sarcomere and look at it up close as contraction happens. And just so you know, this is just showing you, at different parts of the sarcomere, the orientation of the thick and thin filaments, so you can see kind of in different parts of the sarcomere how they would be oriented. You don't have to know all of that, okay? Don't worry about that. Okay, so taking a look here, this is one sarcomere, and let's actually go through the regions of it, and then we'll take a look at what happens when it contracts. Okay, so we see that in one sarcomere, the thick filaments are located right in the center, right? And you can see there's a little bit of overlap here where they overlap each other a little bit. But notice there's this region right in the middle where we have only thick filaments. There's no thin filaments here. So that region where there are only thick filaments, right in there, is called the H zone, okay? You can see it is labeled here, okay? That is the H zone. Okay, only thick filaments. Okay. Then you can see also at the end of the sarcomere, and this will actually cross over the Z disc into the next sarcomere. Notice there's an area where there's only thin filaments, okay? This is called the I band. And if you look closely here, you can see that the thin filaments actually are attached to the Z disc. So Z discs are really an attachment site for the thin filaments. Okay, they're an attachment site for the thin filaments. And so this whole area here at the ends of the sarcomere where we have only thin filaments is the I band. Right in the middle of the thick filament is kind of a thickening. You can't really see it here, but it would be right in the very center of the thick filament. This is called the M line. The M line is where the thick filaments are attached to structural proteins that hold them in place. So the M line is the attachment site for the thick filaments. Okay. The Z discs are the attachment site for the thin. And one more region. Okay, that is right here. You can see there's something labeled as the A band. Notice the A band goes from one end of the thick filament to the other end. So the A band is the entire length of the thick filament. okay, the entire length of the thick filament. As the filaments slide across each other and the sarcomere shortens or contracts, the filaments will actually slide as far as they can. So the thick filament will literally run into the Z discs as the sarcomere shortens. It kind of goes as far as it can until they kind of, can't go any farther. So some of these regions that we just talked about will actually change during contraction. So let's take a look at a contracted sarcomere, and you can see that some of these actually will disappear. You guys have any questions on this? Questions? You guys have that? Okay. And this is just a table that goes over all the parts of the sarcomere. Okay, so this is showing, again, what we just looked at. This is a relaxed sarcomere, which is what we just were looking at. But notice here, the filaments are starting to slide across each other. The Z discs are closer together, okay, because it's shortening. And this is a fully contracted sarcomere. Okay, so notice again, the thick filaments here kind of run into the Z discs. So we have complete overlap of the filaments during contraction. Okay, so in a fully contracted sarcomere, do you see an H zone in the center here? No. So again, there's going to be complete overlap. So this area in the center where we have only thick filaments, it will disappear. Okay, how about the I band? Notice the I band also disappears. And I'm sorry, this is down so low. I know it's hard for you guys to see. But the I band disappears here because, again, the I band is where there are only thin filaments. But as these overlap each other, that will also disappear. So in a fully contracted sarcomere, those are the two regions that actually go away. And we said that the A band is the length of the thick filament. Okay, so it's just the length of the thick filament. Is that going to change during contraction? Does it seem like that would change? No, it doesn't, because they just slide across each other. They don't get shorter, they're just sliding. The sarcomere gets shorter, but the individual filaments do not change length, they just slide. So the A band stays the same, whether it's relaxed or contracted. But these two regions will actually disappear in a contracted sarcomere. Okay, so that is pretty much how muscle contraction happens. Now, we haven't talked about how the action potential gets to the muscle. We've started with the action potential already, traveling along the muscle fiber. So let's just kind of run through those steps. And you guys can review these steps leading to contraction. If you just go through the structure of the muscle cell, it kind of makes you, go through all of those steps that we've talked about. Okay, but if you list the steps leading to contraction, this will also, it's a good way to review the structure of the muscle cell. So kind of vice versa, it works either way. Okay. So we said that the action potential, and I'm just going to abbreviate action potential, travels along the sarcolemma. And what are the tubules called that carry the action potential down inside the muscle cell? the T-tubules. Okay, so when the action potential travels down the T-tubules, what happens next? Calcium is released. So if you get stuck and you're trying to remember parts of this, think about the structure of the muscle cell. We know that T-tubules are located between the terminal cisterns. Terminal cisterns store calcium, okay? So calcium is released from the terminal cisterns. Okay, what does calcium do? It goes out into the sarcoplasm and it binds to, right? Binds to troponin. Calcium binds to troponin. What happens next? What is the part of the thin filament that is going to now move over? We just talked about it. The part of the thin filament that covers actin's binding sites. Tropomyosin, right. So tropomyosin moves. And actin's binding sites are exposed. Okay, and then when that happens, the myosin heads will automatically bind. Okay? Myosin binds. And as soon as myosin binds, there's that kind of pivoting motion where the filaments will slide across each other. And that's what contraction is. Okay, so this is the actual contraction process when filaments are sliding. Okay, so the only thing we need to include now is how does the action potential get to the muscle fiber? Our muscles don't just decide to contract on their own, right? They have to be told by the nervous system. So we're going to talk about how a neuron sends the action potential to the muscle fiber. Do you guys have any questions on any of this before we go on? Anything at all? Don't be shy. Uh-huh.
So does it slide and then that it binds, or does it bind like pull?
Yeah, so what happens is the myosin cross bridges, do this very fast, of course. So they're constantly binding and releasing, bind, release, bind, release. And so it's a smooth process. It's not jerky, like I'm showing it. It's a smooth process. But this continues as long as there's calcium binding to troponin. This will continue. So to allow the cell to relax, calcium actually has to be pumped back into the sarcoplasmic reticulum, which is actually where the muscle cells use a lot of their ATP. It's estimated that muscle cells use about 40% of their ATP just to allow the cell to relax. Okay, so it's kind of funny because we use energy to cause relaxation, but the calcium has to be removed from the sarcoplasm and put back into the terminal cisterns. So that's something that's constantly happening, and it's called the calcium pump. But until then, the cross bridges are constantly moving. They have their own, they store their own ATP. The little myosin heads can store their own ATP, and they store the enzyme that breaks it down so that they constantly have ATP to power that movement. Is that what you were asking? Okay. You guys have any other questions on any of that? Okay. So, You may have heard of the different types of neurons that we have. This is what we'll be talking about more in the next unit. But basically, we have two types of neurons. We have what are called sensory, which I know you guys have all heard of. The other type is called motor. Motor meaning, you can think of that as having to do with movement. So this is the actual definition of a sensory neuron. These are neurons that carry their action potentials or electrical currents to the central nervous system. You guys know CNS is central nervous system, right? Brain and spinal cord. Okay, motor neurons are the opposite. They're going to carry the action potential away from the CNS, away from the brain and the cord, out to our muscles, okay, telling the muscle to contract. The actual definition of a motor neuron is a neuron that carries the action potentials away from the CNS to either muscle, tissue, or glands. Okay, we're not talking about glands right now, but I wanted to include this just because this is the actual definition of a motor neuron. Okay. So we're going to take a look at what a motor neuron looks like and how it kind of connects to the muscle fiber. Okay, so this is a diagram that's actually from chapter 14, and I want to just simplify this a little bit, but you can see this is kind of a little bit more detailed. That's more what the real thing looks like. Motor neurons have all of these kind of processes that are attached to the cell body. Wow, this is not coming out well. Let me start over here. Okay, so they look something like this, and here's the nucleus of the cell. So this would be the cell body. This is going to be the one long process that usually branches at the end. And then we have these little kind of knob-like structures here. Okay, so again, this is a motor neuron. So motor neurons have many processes attached to the cell body called dendrites. Anyone know what dendrites do? They receive incoming signals, right, from other neurons. Okay, receive incoming signals. So they receive information coming in from other neurons. If this neuron is going to send an action potential, then it will send it along this process here. What is this process called? This is the axon. Okay, so all neurons have just one axon, and that sends the action potential. Okay, so if this neuron decides it's going to have an action potential based on all that incoming information, it will start, that electrical current will usually start right here at the beginning of the axon, and it will travel all the way down to the end of the axon. Okay, so this is the direction the action potential will travel. So this is just like in muscle cells where the electrical current is literally just ions, atoms that have an electrical charge crossing over the cell membrane, but it moves in a wave-like pattern. So it will start here and it will travel down to the end. Okay, so notice at the end of the axon, we have these little knob-like structures. These are called synaptic knobs or synaptic bulbs, and they contain what is called the neurotransmitter. Okay, so synaptic knobs or synaptic bulbs. Okay, and although this shows them, it's kind of hard to see them here, but that is what we are talking about, these synaptic knobs. Okay, questions on? Mm-hmm. Okay, so you guys have probably heard the term synapse before. We know that synapses are connections that neurons make. Maybe with another neuron, or it could be with a muscle fiber, it could be with a gland. It's just a connection between a neuron and another structure. Okay, so this is the definition of a synapse. It's just the junction or connection between a neuron and either another neuron or a muscle fiber or a gland, whatever it happens to be synapsing on. Okay, so we're gonna talk about this special type of synapse between a motor neuron and a muscle fiber. And this is called the neuromuscular junction. You can also turn that around and say myo. You guys know myo refers to muscle. Myoneural junction, same thing. So this tells you it's between the neuron and the muscle fiber. Okay, so just a special synapse between a motor neuron and a muscle fiber. Now you do have a picture of this in your book, but this is one of those things that I like to draw because I think it's helpful in understanding the parts of it. So let's take a break, and when we come back, we'll talk about the neuromuscular junction, okay? So let's come back at 8:40, and we'll go over that. And we are almost finished with muscle tissue. Okay, this is gonna kind of wrap up chapter 10. Okay, so come back at 8:40.